Hi, everyone. Welcome back to the afternoon session. I hope you've had a nice lunch break. This afternoon session will be chaired by myself. I'm Angela Wu from the Hong Kong University of Science and Technology, and my co-host here.
So my name is Natini Jinawat from Mahidol University.
Okay, and this session we're going to have the non-infectious disease and cancers sections, and I'll be introducing the first speaker who's already online and waiting for us. So we have as our first speaker Dr. Zeming Zhang. He is a professor at BioPIC in Peking University, and he has been a long-time researcher in the field of single-cell tumor microenvironment studies. So I believe he's published a couple of really nice papers recently on this topic, and he'll be sharing this with us today. So without taking too much time from Zeming, please let's hand over the stage to him.
Good afternoon, everyone. First of all, it is my pleasure to be included in this strong speaker lineup. Secondly, I wish I were giving this talk in Thailand. I've never been to Thailand, so I would try to get invited again in the future. Today, I will present our studies on pan-cancer analysis of the tumor microenvironment. The concept of the tumor microenvironment is not foreign to most people. It is essentially the collection of various cell types making up the entire tumor mass. But there are still many unknowns about the TME that hinder our understanding of the interplay among all the cell types inside the tumor. Over the last few years, my lab has been working on understanding the special features of TME, ranging from its composition, cell fate, special functions, cell interaction, spatial features, to other properties like heterogeneity. Today, I'll focus on the heterogeneity aspect of the TME. So why do we want to study a TME heterogeneity? Because TME likely will impact many clinical parameters, including how patients respond to immunotherapies. As we know, although immune checkpoint blockade therapies, such as anti-PD-1 antibody treatment, can cure some of the cancer patients, the clinical response rates vary dramatically depending on the cancer indication. There are some existing predictors for such responses, such as the tumor mutation burden, or TMB, or the PD-L1 expression levels, but they are not very accurate. We think that the immune properties within the TME might be more useful. Our earlier study focusing on infiltrating T cells have shown that the infiltrating T cells in colon cancer, liver cancer, lung cancer vary dramatically, even though at the blood level, the differences is actually minimal. So we think that it is important to study the extent of such differences across different cancer types. In addition, some of the meaningful cell types are only found in one particular cancer types. For example, in our own study early on, we found the LAMP3DC from liver cancer, and from colon cancer, we found the SPP1 TAM and the C1QC TAMs, or tumor-associated macrophage cells from colon cancer. It's not really clear if these cells are unique to a particular cancer type or if they are universal across all cancer types. There is only one definitive way to address this question, which is to compare infiltrating immune cells across a variety of cancer types, hence the pan-cancer analysis of the TME. So in this case, we constructed a pan-cancer single-cell transcriptome atlas for myeloid cells. We downloaded all possible published single-cell data sets for cancer-associated myeloid cells and generated new data when needed. So these are the major cancer types we covered. And for each of the cancer type, we would analyze them separately and try to identify all possible major clusters. And for each of the cluster, we would try to identify subclusters. And here's just a simple example showing the results for esophageal carcinoma. And then you can repeat this process for all major cancer types. So we're able to consistently identify cell types from monocytes, dendritic cells, and we are able to identify LAMP3DCs across all cancer types, indicating their universal role across all cancer. So here we found LAMP3DC in all cancer types. But even in this case, even though qualitatively they are present, if you look at the relative proportion of such cells within each cancer types, they vary dramatically. Nasopharyngeal carcinoma and stomach cancer, for example, have a very high level LAMP3DC, as opposed to breast cancer and kidney cancer. By contrast, some other cell types like CDC2 have very little variations across cancer types. Some other cell types like SPP1 macrophage cells are only found in about half of the cancer indication, but not others. So clearly there is a big variation of tumor infiltrating immune cells, at least myeloid cells across cancer. How about T cells? So using a similar approach, we constructed a pan-cancer single T cell atlas by combining all possible published data sets, and we supplement those with our newly generated single T cells. At the end, we collected data for 316 donors representing 21 major cancer types. After quality filtering, we obtained data for about 400,000 T cells. So skipping all the technical details, we obtained a total of 41 metaclusters, and each of this metacluster has its unique distribution properties in tumor, adjacent normal tissue, and it is obvious that both exhausted T cells and the activated Tregs have very high abundance in tumors, but low elsewhere, including adjacent normal and blood. We found that there is a large variation of T cell subtype abundance. Here are just a few examples. The terminally exhausted T cells could be found in all cancer types, but their abundances vary dramatically. The median frequencies of terminally exhausted T cells are highest in esophageal carcinoma, liver cancer, and head and neck cancer, but they are barely detectable in multiple myeloma, basal cell carcinoma, and breast cancer. And the tumor-activated Tregs generally have high frequencies, but still exhibit high variabilities. For example, in esophageal carcinoma, head and neck cancer, the frequency of such activated Tregs are twofold higher than those in breast cancer and stomach cancers. Okay, so if you have a strategy targeting a particular cancer type, such as activated Tregs, it's meaningful to select the right patient indication, cancer indications, where the such T cell phenotype is a notable feature for this cancer type. So those are the published work, and I'm going to present a few slides on unpublished work. So in addition to T cells, we also recently performed a pan-cancer analysis of NK cells, or natural killer cells. Given that within the tumor tissues, the NK cells are relatively rare among CD45 cells, and that their heterogeneity is still poorly understood, it is both difficult and important to build such a large-scale NK cell atlas. Overall, from a total of 71 data sets, we collected 160,000 NK cells from over 700 donors representing 24 cancer types. So as you can see here, the proportion of NK cells among cancer types vary dramatically. Cancer types such as cholangiocarcinoma, ICC, the kidney cancer, liver cancer, have very high abundance of NK cells overall than the NPC, colon cancer, and esophageal carcinoma. NK cells are typically classified by two conventional markers, CD56 and CD16. And there are two major groups of NK cells, the CD56 high, CD16 low cells, which are abundant cytokine producers, and those CD56 low, CD16 high, they call it dim and high, dim and blood bright. Okay, so these cells are highly cytotoxic. But if we examine the single cell data we collected, there is further heterogeneity within each group that are not known before. For example, those CD56 high, CD16 low NK cells do not always express known cytokines. These are the high expressors, but these are not high expressors. Similarly, not all CD56 dim CD16 high NK cells have the same cytotoxic properties. And some of the cells may have high expression in cytokines that can be used for recruiting other cell types like CCLs and 3 and 4. Since this is not an immunology meeting, I'll skip why these expression patterns are meaningful. It is suffice to say that different subsets of NK cells have different properties associated with cytotoxicity, inflammation, stress, and cell recruiting capabilities. Accordingly, different types of tumors exhibit remarkable differences regarding their NK cell composition subtypes, as shown here and here. For an extreme example, the NK subset composition are completely different between nasopharyngeal carcinoma and pancreatic tumors. This may explain some of the controversies observed in the NK study field. For example, does a double positive NK cells exist? Well, it depends on which cancer types you happen to be working on. If you work on gastric cancer, liver cancer, and kidney cancer, you probably will never find any CD56 high and CD16 high cells. By contrast, if you work on melanoma and prostate cancer, we likely find a lot of double positive NK cells. So similarly for other NK subsets that I will not elaborate, there's also high degree of heterogeneity. So what does this all mean? Could the tumor-infiltrating immune cells define cell types? So we're doing such studies now, but here's just only a proof of concept work that we did with T cells only. So in this case, we said we only took the T cells. We ignored all the other cell types, including the cancer cells. I asked the question, can we classify cancer type based on the T cell compositions only? And since we can, we are able to classify all cancer, over 316 patients, into two major groups and about 8 different clusters. Two major groups are generally have high exhausted T cells, low presence of resident memory cells. The other group has the opposite patterns. Okay. But then if you look at the 8 different groups here, they have different enrichment for cancer types. For melanoma, for example, over 50% of cells fall into the C2 category here, which has higher presence of exhausted T cells, but low presence of activated Tregs. This may indicate this is a good group that respond to ICB treatment. By contrast, thyroid cancer has a lot of C3 groups here, which doesn't have a whole lot of exhausted T cells, but has enriched interferon positive TRM cells here. Okay. So then we ask the question, if you take this classification, would this classification correlate with responses to immunotherapy? And it seems like there is a trend they would correlate. If you take, there's not a whole lot of data for those, but if you take those melanoma sample treated with anti-PD-1, that seems like the responders tend to fall into this category where you have lower exhausted T cells and high TRM groups of cells. Okay, so at the end, what we hope to do is that we take all those data together, we integrate all the data so that we can classify cancer using the immune properties alone, so that we can beat the current golden standard, which is a tumor mutation burden. So I'm running out of time, just want to mention that we observed a lot of tumor microenvironment differences across cancer types. This indicate that there should be a path forward for personalized immunotherapies. So to finally acknowledge many people who contributed to this study listed here. I'm running out of time, so I'll just leave this up for a few more seconds. Thank you very much.
Thanks, Zhoming. Very nice talk as usual and perfect timing. So we have time for questions. Any questions? Oh, I see Shyam there.
Hi, Zeman. very exciting tour. As usual, you're really a pioneer in pan-cancer immunoanalysis. I'm curious, and the last thing you said was very tantalizing. So do you have a short answer for, are you able to beat the tumor mutation burden prediction from your existing analyses?
The short answer is yes, but it's not based on all the cell types. So what we have tried is this. So we found out that two subsets of T cells are fairly important. And those are both CD4 and CD8 T cells that highly express the gene called CXCL13. So each of the group of cells seem to be tumor-reactive T cells. So if you take the relative abundance of those cells and you only factor those two cell types, you can actually beat the performance by the TMB. So the tricky part is that this is still based on the tumor analysis, not a peripheral blood sample. So what we are doing now is to try to correlate the kind of cell types or some other indicators in a peripheral blood that seem to be correlated with those tumor-infiltrating immune cells. So those are the kind of analysis we're doing. So what we try to do these days now is to identify the most important immune cell types that are pertinent to the immunotherapy efficacy. So far, I think we have a few handles, but to turn those into more clinically useful biomarkers is still going to take a little bit of time.
That's exciting. And do you know the TCR clonotypes of those cell types?
Hello. Sorry, I lost a connection for a few seconds.
Yeah, just repeat the question.
Do you know the TCR clonotypes of those subtypes?
Actually, yes. So the way to identify if the cells are tumor reactive is to identify exhausted T cell first, and those presumably are the cells recognizing tumor signals. Then use a TCR as a handle to probe other T cells. You can find other non-exhausted T cells that happen to have the same TCR pairs. So those, so you can definitely find the TCR information through this approach.
Brilliant. Thanks.
Okay. Maybe a quick one from Woongyang over there.
Okay. Hi, Jamin. Actually, did you have a chance to compare with the TME subtype based on the bulk RNA data? I mean, there's some 4 subtypes based on TME features, and then immune-enriched and immune-enriched fibrotic something.
Right. So this is based on the colon cancer study, right?
Yep.
Yeah. So I believe we did for so since this study is not our study is not focused on the colon cancer, so this only applied to a subset of the. I believe we based on our limited data sets, because our data sets is a much smaller data sets than the TCGA data sets. So we're able to find some of the subtype you just described. So, but I don't think the systematic comparison has been done. So I understand you probably have a lot of useful data sets that can allow you to thoroughly answer this question. But in our side, I think for our own colon cancer data, we're talking about only like less than 20 tumors to work with. So that wouldn't be a good data set to definitively answer this question.
Okay. Thank you.
Okay. Unfortunately, we don't have more time for questions. I saw a couple other questions in the room, but I'm sure Zeming would be happy to answer your questions on email as well. So thanks, Zeming, for joining us remotely.
Thank you, Angela.
Bye. Okay, before I introduce the next speaker, I also wanted to mention that actually there is a third session chair who unfortunately also could not be here today, Fu Choutang. So Dr. Fu Choutang, he's actually at Peking University with Zeming, and it's too bad that he couldn't be here today. So I just wanted to acknowledge that he also put in a lot of work to organizing this session. Okay, so on to our next speaker for the session, Dr. He Mu. So she has recently joined us in Hong Kong, not at my university, at University of Hong Kong, from UCSF. So she previously contributed her work to the tabula muris, consortium, and she's also constructed a single-cell atlas of mouse trachea to study development of cells in the airway. Although this session is on disease and cancer, but she's actually going to be talking about normal tissue, I think. So welcome, He Mu.
Good afternoon, everyone. Thanks very much for the invitation to present our work here. I apologize that I couldn't be there in person due to some last-minute COVID situation, and I very much look forward to seeing everyone in our next meeting. So my lab at the University of Hong Kong is working on tissue regeneration and repair. In particular, we're focusing on the respiratory tract as one of the major mucosal barrier that separates our internal organ from the external environment. So when we're born and start breathing, we start to interact with the natural and industrial environment. The air would travel through the canal-like structures and reach the distal lung for gas exchange. And the cells that lines the mucosal barrier are very important for all the physiological and pathological conditions that we have to deal with throughout our lifetime. So the cells that cover our respiratory tract are primarily divided into three types. The basal progenitor cells, which will give rise to ciliary cells that have the motile cilia axoneme projecting on the apical surface, and the secretory cells that form the secretory granule and secrete mucin. And these ciliary cells and secretory cells would form a very intricate engineering system called the mucociliary clearance to transport the mucus. So we can visualize the cilia beating. So for example, this is a newborn mouse airway, and you can see these tiny cilia that can generate a constant beating behavior. And we can also visualize the mucus flow. So if you add the fluorescent tracer particles into the airway, these tracers would attach to the mucus, and you can see this coherent and unidirectional flow from one end of the airway to the other. So using these combination methods, we were able to visualize the action and the behaviors of these ciliary cells and mucus, secretory cells, and to really study how these cells function at the mucosal barrier for protection. It turns out these cells are actually highly evolutionarily conserved. The ciliary cells are present in many animals throughout the animal kingdom. So, for example, in this flatworm, the ciliary cells cover the ventral surface of the planarian, and the cilia are required for general motion and gliding. And also you will see lots of ciliary marine larvae. The cilia are formed on the outside of these small creatures, and they're required to swim, to glide, to feed. So essentially, it's very interesting to think about how these ancestral cell types would be so important for different kinds of animal physiology, but also they're able to cover the respiratory tract, which is a relatively new evolutionary invention in all the air-breathing animals. Not surprisingly, these cells are also the target for many kinds of respiratory diseases, including the one that caused by genetic mutations, such as primary ciliary dyskinesia or cystic fibrosis. They're caused by complex immune environment interactions, such as asthma and COPD, and also caused by diseases caused by infections of bacteria, virus, and fungi. For example, SARS-CoV-2, the virus that caused COVID-19, would primarily target these motile ciliary cells before they invade the human body. So the question that we're trying to address in our lab is, how do these ciliary cells differentiate in the mammalian airway? And how do they function to protect the airway, to generate the flow, to perform this defense function at the airway? So if we're looking at this mouse embryonic airway on the left and the newborn mouse airway on the right, I hope you can appreciate that the very dramatic difference in terms of morphology. So during embryonic development, all the progenitor cells look essentially the same. But just within a few days, these progenitor cells would differentiate into morphologically and functionally distinct cell types. So how are they differentiated? Is everyone created in the same way? And within the same population, is there any sort of functional and behavioral heterogeneity that would drive their distinct function in physiology and pathological conditions? So to address these questions, we started to construct an airway atlas that would expand the critical time points during differentiation and transition from the amniotic fluid to air, so basically at the neonatal stage. In collaboration with the CZ Biohub, a few years ago, we collected mouse airway samples from 2 time points during development and the postnatal day 1 and 2 after the animals are born. So we collect all the cells from the airway, the major airway, and build the cell atlas. And we could actually see all the essential cellular components from the atlas, including the epithelial cell types, immune cells, endothelial cells, neuronal cells, and all kinds of flavored fibroblasts. So all the cells would play distinct functions in the airway physiology. And we were also fortunate to get 2 human fetal samples from gestation 21 to 23. So we were able to actually also sequence these human fetal airways and can combine and compare the cell types and cell states between mouse and human during a rather similar developmental stages. So when we put together all the data between mouse and human, we're able to see that the mouse and human share very conserved cell types for the apical, the surface epithelial cells, and also the mesenchymal cells. These cell types can be identified by very similar gene markers. The major difference between the mouse and human airway is that human has large amounts of some mucosal glands. These glands are secreting a large amount of mucus, presumably because human airway is so much bigger than the mouse. In the mouse, the submucosal glands are very limited and restricted to the proximal of the airway. So using these comparative atlas, we can really start to understand the cell type difference and similarity between a mouse and human. And we can also try to study some aspects of human disease using mouse models, given that the cell types could perform very similar functions. So for today's talk, I want to focus on the ciliary cells, because we believe that that's a very important cell type within the respiratory tract. So as I mentioned before, the ciliary cells are primarily differentiated from the progenitor cells. And we can identify these cells using immunofluorescence staining or gene expression profiling based on the expression of transcription factor FOXJ1. So if we look into our data set, when we combine the embryonic ciliary cells and the mature ciliary cells, all of the cells would express FOXJ1, and that's how we identify the cells. But if you look at the overall transcriptomes, the immature and the mature ones are actually quite different. So there are a lot of genes responsible for the structural components and the construction of cilia are highly expressed in the immature stage, whereas in the mature stage, a lot of the markers that are responsible for signaling are regulated at that stage. So we believe that there are very distinct stages for structure and function that are expressed at different stages. And even if we zoom into the immature stage, there's still a lot of heterogeneity within all the cells before the animals are born. So if we isolate the immature cell cluster and increase the granularity for cluster analysis, we could actually see 3 distinct subclusters. At that time, we're just trying to use the conventional RV velocity analysis to really ask the question, what's the trajectory relationship? What's the spatial relationship during development? Do these cells would these cells exhibit? So based on RNA velocity analysis, we came up with this very stable lineage relationship in which there are 3 clusters, and cluster 1 would give rise to cluster 2 and cluster 3. A lot of the genes enriched in cluster 1 are also being studied in using model organisms to implicate it in cellulogenesis. So we believe that this cluster 1 would function as a precursor state. And it turned out that this cluster relationship is very stable. In collaboration with Yuanhua Huang, also at University of Hong Kong, we're able to test with a bunch of other velocity analysis tools, and all of them came back with the same trajectory prediction, in which the precursor state would give rise to the more mature states. So we really believe that for cilia development, this would be a very stable trajectory. And we're hoping to use different tools to really confirm that. And also, we were happy to see that even in human samples, these trajectories are highly conserved, and we can see using the fetal samples and also different atlas that the precursor states are highly enriched in the human embryonic development. And a lot of the gene modules enriched in the states would be very important to understand the human ciliary primary ciliary dyskinesia symptom. So one thing that's very important that came from our analysis that we noticed that there's a ciliary secretory hybrid cells in the neonatal mice. They were not there during development. We haven't seen any similar reports for the adult mice, but we can actually see very distinct gene expression pattern in the hybrid. So the hybrid cells would express markers for ciliary cells. They would express marker for secretory cells. And we also were able to verify the cell states using orthogonal conformations, including transmission EM, in which that we can see the mucosal granules and the motile ciliary projections present in the same cells. So we're very happy to identify this new cell states in the neonatal stage, and we thought this could be some cell states that are important for a unique physiological condition. But it turns out that they're actually also present in human disease states. So, for example, before COVID-19, 2 papers report the presence of hybrid cells in human nasal polyps that caused by chronic inflammation, and also in asthma patients. And in one of the human COVID-19 atlas, we also notified that there's this FOXN4 positive cells. The cell cluster is kind of bridging the ciliary cells and secretory cells, and this cluster also expressed very similar transcriptome profiles at the hybrid cells that they found in the asthma patients. So now the question is, obviously, there's a lot of cellular heterogeneity and plasticity, so what could be the cellular origin for these hybrid cells? It's very difficult to prove that in human samples, so we thought that our mouse models, given that we have observed this cell state in the neonatal mice, we could actually test that using the conventional lineage tracing analysis. So using 2 different methods, so one can trace the ciliary cell lineage, the other can trace a secretory lineage, where we're able to perform lineage tracing using the fluorescent marker. So it turns out at the neonatal stage, the hybrid cells are predominantly coming from the ciliary cell lineage, as a ciliary cell tracer could show that some of the traced cells also express the secretory marker. But also, if we use the secretory cell lineage tracing, we could also see some of the traced cells express the hybrid cell markers. So that could suggest that even during a defined physiological condition, the cellular origins for hybrid cells are not the same, right? So different lineage could all contribute to the hybrid cell states. And we also use a ciliary cell lineage to ask the question whether in an induced inflammatory condition, can we see the transition, right? Because in the human asthma condition, the inferred trajectory for these hybrid cells is the ciliary cells. So we test the same kind of induced inflammation using mouse models. And we try to analyze the presence of hybrid cells using the trace cells. So it turned out that some of the cells still maintain their cilia apparatus. They do not express the secretory protein. But in some of the trace cells, we start to see losing of the ciliary marker and upregulation of the mucus. So these experimental results would suggest that perhaps similar to the neonatal stage, the ciliary cells could contribute to the hybrid cells in the induced inflammatory condition. But perhaps there's also a level of heterogeneity in terms of cellular origin. So now we can put back the cellular origin and combine with different results from other groups that it seems that this plasticity is highly dependent on the specific cellular stimuli. So in some conditions, the ciliary cells would contribute to hybrid cells, but in some other conditions, perhaps the secretory cells or basal progenitors could also differentiate into hybrid cells. And the next question is obviously what would be their cellular function? And that's a more complex question that we're hoping to address in the future. So now when we look back onto this picture that the embryonic cells would all have the progenitor cells, whereas the newborn cells would exhibit very differentiated cell types. And we can look into their atlas, we can combine different atlases. So basically having a complete system help us to point towards the thing that we don't know and to recognize a part of the elements that cannot be explained by the parts we understand. And hopefully combining with all these animal models and different atlases, we can start to better understand the level of heterogeneity and plasticity of these important cell types for development and differentiation. And also wanted to point out that we actually need to develop a more human-like system to understand human biology, because these cells are highly abundant in mouse, but they also they're very conserved in humans, so we start to differentiate human organoids. So hopefully we can one day address all these hypotheses that we have verified in animal models in the human-like structure. And I also want to point out, because these cells are highly conserved, so we should really combine different knowledge from different species, from Plenary, from the trypanosome, from all these animals that have the motile ciliary cells, to really appreciate the cellular complexity and the transcription regulation and the plasticity of the cells to better understand human development and disease. And last, I would like to acknowledge my previous mentor at UCSF, Lady Jen and Yunong Jen, who really supported a major part of this work. My collaborator Spiros and Bing, who were used at the CZ Biohub and now at Genentech. My collaborators who provided the human samples, and also my current collaborator Yuan Hua and Hong Kong Yu, who were working together with to really look into more details of the trajectory. So I will also like to thank my lab, these fearless young people who joined my lab and really trying different things. And obviously we're expanding, we're trying to recruit. So if you're interested, we look forward to hearing from you. And again, thanks very much for the invitation. I appreciate your time, and I'm ready for questions.
Okay. All right, thank you, Mu. So are there any questions for Dr. He? Oh, I see, is that a question or? No, not a question. Any questions? Okay, so I have one quick one maybe. You mentioned the epithelial, I guess it's the air-liquid interface organoid model. I think that's a great idea, but I've also seen a little bit of data which is unpublished showing that the composition of cells in the organoid model could be quite different to that of in vivo. Have you found that, or do you have any thoughts on maybe how the model could be improved if that's the case?
Yes, definitely. I think the major challenge in the field is that how do we push the cell states into a more mature state, right, like the adult human being? Because we're using iPSC and ESC derived organoid models, and these cells actually, the differentiated ones would look more similar to the neonatal stage. And I think if you get stem cells from the tissue, then maybe it's easier to get some cells that are more mature. But in general, we're trying to again, I guess the field and the entire field is trying to build models that would be more mature metabolically and morphologically, and also have a more complex cell type that would support a function.
Mm-hmm. Okay, awesome. Yeah, I will talk to you more about the organoids when I get back to Hong Kong. All right, hope you can join us next time. Bye.
See you.
This is next week. Okay, Thank you, Angela. Okay, so let's move on to our next speaker. So Dr. Dean Tham Kieu, okay, so is an associate professor at the Department of Drug Discovery Medicine and the Graduate School of Medicine, Kyoto University. So his research interest is in the development of novel drugs for allergic and autoimmune disorders and cancers. He's also a fellow Thai researcher. So please welcome Dr. Dean.
Okay, thank you very much for your kind introduction. So my name is Dean Tham Kieu. So I'm from Department of Drug Discovery Medicine, Kyoto University. The topic of my talk today is PG2, EP2, EP4 signaling mediates immunosuppression in tumor through the facilitation of MREG-DC Treg axis. So first of all, I would like to give a brief introduction to you guys for who do not know very well about PG2. So PG2 is a lipid mediator. It's generated from the lipid, the fatty acid, arachidonic acid, through the catalytic activity of COX and PGE synthase enzymes. It was previously known that there are 4 cognate PG2 receptors, EP1, EP2, EP3, and EP4. And previous research results by us and another group, we suggest that EP2 and EP4 is highly expressed in tumor infiltrating immune cells in different human cancers. So based on this background, several pharmaceutical companies, they made the EP2 and EP4 antagonists and put those drugs into the clinical trial. But unfortunately, because the basis or the mechanism of a PG2 signaling in cancer is not very well understood, none of those, well, at least none of these clinical trials has been yet a success. So we started our project, try to dissect the mechanism of a PG2, EP2, EP4 signaling in cancer, particularly in, well, the cancer immunity. We start our project by test or examine the effect of EP2 and EP4 antagonists in the mouse cancer model LLC1. We chose LLC1 because this model it is insensitive to the checkpoint inhibitor, including anti-PD-1 antibody. As you can see in this graph, so in compared to the control vehicle, which is shown in the black line, the EP2, EP4 treated mice has much smaller cancer growth in compared to the control. It's also very clear if you take a look at this picture down the side here. So it results in about 70 to 80% suppression of the tumor growth. So we are wondering about the mechanism here. So to dissect the mechanism, so we decided the next experiment to test the tumor growth of the wild-type LLC1 tumor cells in wild-type mice, wild-type control mice, or in the EP2, EP4 double knockout mice. So in this case, we transplant the wild-type cells into the control or into the double knockout mice, and then we found again that when we transplanted the LLC1 tumor cells to the knockout mice, the double knockout mice, it shows again the tumor growth suppression effects. So what does it mean? So it means that, well, the tumor suppression effect of the EP2, EP4 suppressions is mediated by the cell in the host, not the cancer cell itself, yes. So it means that it might be mediated by the stromal cell or by the immune cells. But because, well, in the previous study, they suggest, well, the expression of EP2, EP4 in the immune cells. So we speculate, or we hypothesize that it might, the effect, tumor suppression effect mediated by EP2 and EP4 might be important in the immune cells. So we next conduct the single-cell RNA sequence analysis of EP2, EP4 antagonist-treated tumor-bearing mice. As all of you guys know very well about this, so I'm going to skip this and just tell you the results. So we finally obtained about 32,000 tumor-infiltrating immune cells transcription profile from 6 tumors. And then the unbiased clustering analysis results in 15 different subtypes of LLC1 tumor-infiltrating cells, as shown here on the right side. And then we annotate the name of the cell subtypes here, 15 cell subtypes here, and then, the name is shown here as according here. So we have, in this model, we have, what is special about this model, we have very few lymphocytes and we have a lot of myeloid cells. Because of the limitation of time, I cannot introduce you all of the analysis results. Today, I'm going to just focus on the analysis results of the cell we call mRegDC. So for you that is not so familiar with mRegDC, so I would like to give a very brief introduction about what is it. So mRegDC stands for major DC enriched in immunoregulatory molecules. So actually, it was in the literature. It was first, I think, it was first reported by Zemin in his paper in 2019. But this paper by Miriam Murat group, I think this is a paper that the paper that they established very well about what is the mRegDC. So basically, the DC, well, as you know, it's an immune activating cell, so it presents the antigen to the T cell and activates it. But this mRegDC cell is special because, well, in contrast to the other DC, it expresses a molecule that is immunosuppressive, like PD-L1, FAS, IL-4, I1, and then CCL17 and CCL22. Actually, among the tumor-infiltrating immune cells, they express this molecule at the highest level. And to our surprise, when we conduct the differential expression genes analysis, the DEC analysis on mRegDC, we found that upon the treatment of EP2 and EP4 antagonists, the chemokine CCL22 was the second most suppressed gene. And then, given that previous study suggests that CCL22 and its homolog CCL17 works as a chemoattractant for Treg, we speculated that it might affect the Treg tumor infiltration. And to test that, we conduct a FACS analysis of the tumor infiltrating Treg cells. So the red bar represents the number of the tumor infiltrating Treg cells in the EP2, EP4 antagonist treated tumor-bearing mice. So you can see, in compare with the white bar of the control, the number is significantly suppressed in the EP2, EP4 antagonist treated mice. So our conclusion is that EP2, EP4 antagonist treatment suppress the tumor infiltration of Treg. So this is the final conclusion for my talk today. So I think that, well, in the tumor microenvironment, PGE2 is highly produced, and then it exerts its function through the EP2, EP4 receptor on mRegDC, and then it promotes the CCL22 production. And in combination with CCL17, it recruits the Treg to the tumor, and because Treg is a highly and very potent immunosuppressive cell, it results in the tumor immunosuppression microenvironment. This is my final slide. So here is shown my collaborator. So I would like to use this opportunity to say my special thanks to Dr. Wardom, who is the organizer of our meeting today. Thank you very much.
Thank you for a great talk. We now have time for a few questions. Okay, so I have one question for you. Have you explored into the roles of NSAID, in dealing with this PGE2, and then do you think there is a role of using NSAID as more like repurposing anti-cancers, something like that? Yeah.
Yes. So due to the limitation of the time today, I didn't present the data, but actually I also treat this LLC tumor-bearing model mice with the COX-1 inhibitor, COX-2 inhibitor, and also aspirin. So we found that COX-1 suppress about 50% of the tumor growth, and then COX-2 also suppress about 50% of the tumor growth in this model, and then in combination it shows a synergistic effect, and it's almost equal to this EP2, EP4 antagonist in combination. Yes, and aspirin also show a similar effect to our EP2, EP4 antagonist tumor
Okay, very
suppression.
interesting. Yeah. Okay. So any other questions? Okay, so thank you so much. If not, we will just move on to our next speaker.
All right, so we're now on our final speaker for this session, and we have a junior trainee to give this talk. So her name is Yichun Elaine Tao, and she is currently doing her PhD in quantitative biology and medicine at Duke NUS. And I think she's going to share with us her work on bioinformatic analysis of dysfunctional CD8 T cells. Take it away.
Yeah, okay, thanks for the kind introduction and giving me this opportunity to share some of my PhD work here. So my topic today is single-cell data analysis for the evolution of CD8 T cell dysfunction in head and neck squamous cell carcinoma. HNSCC is the second most common cancer worldwide and is a very aggressive cancer type. For the treatment, apart from the traditional therapies, immunotherapy nowadays provides another option for cancer treatment. And in HNSCC, studies show that compared to the standard therapy, PD-1 blockade have increased the overall survival from 16% to 36%. And however, around 85% of patients with metastasis has no response to PD-1 blockade. Therefore, we focus on one specific tumor sample, immune cells, which is CD8 T cells, and it is a major driver of anti-tumor immunity. So this a review paper summarizes current knowledge of CD8 T cell states from a wide range of different cancer types. In the tumor microenvironment, chronic and continuous antigen exposure drives the CD8 T cells from the naive-like state to a different degrees of dysfunctional states. And along this axis, cells acquire certain feature changes, including the inhibitory receptor expression. Can you see my mouse? Sorry. Never mind. And the increasing of CXCL13 expression. And also there's a burst of proliferative capacity near the early dysfunctional axis. Indeed, these dysfunctional CD8 T cells are a major cell type that are responding to immunocheckpoint. However, the very late dysfunctional stage cells may have limited response to immunotherapy. Therefore, utilizing single-cell RNA sequencing, we established the trajectories for T cell dysfunction and discover the key regulators that could alter the T cell dysfunction. We collect the samples of paired oral primary and lymph node metastasis HNSCC tumor from 7 patients, followed by 10x single-cell RNA and TCR sequencing. In total, we identified 13 major cell types, and for today, we'll focus on the T cell cluster, and specifically to extract the CD8 T cells for reclustering. Then, based on the canonical markers, we identified 6 different states of CD8 T cells in our patient, including naive-like, memory, pre-dysfunctional, proliferative, and late dysfunctional clusters. As expected, most of the naive-like cells are from the lymph node metastatic sites, whereas the primary cells are more towards the dysfunctional states since they exposed to the tumor antigen for a longer time. Then, based on these clustering results, I applied slingshot to infer the trajectories. And using the late dysfunctional cluster as the endpoint, slingshot identifies one trajectory starting from naive-like state. This naive to dysfunctional transition is supported by the changes of known features. And on the right, these score plots showing that along the pseudo time, the cells are gradually lose the naive markers and acquiring dysfunctional markers. And there's also a bump of proliferation, which likely reflects expanding clones for tumor-targeting CD8 T cells. And also, we observe CXCL13 elevation at the end of the trajectory. So this plots here will replicate the feature changes described in the review paper I mentioned early on. So next, to further investigate the novel regulators along this dysfunctional evolution, we developed this computational tool called gene switches to identify the switching genes along the pseudotime. Gene switches is a statistical framework that processes gene expression matrix together with a pseudotime ordering to find the set of genes that switch during the transition. There are three main steps involved here. For each gene, we first binarize the gene expression into off and on binarized state. And next, we fit a logistic regression model on this binarized data to identify the switching time point for each gene. Lastly, we plot the significant switching genes along the pseudotime for visualizing the orderings with up and down regulation, and the y-axis representing the quality of fitting. And so there are some highlights of gene switches. It discovers the genes that have clear on or off switching patterns, which can be good candidates that drive cellular transitions. And also, since we use the binarized data, it can fast fit the logistic regression model, even on the large data sets. And also, it provides robust ordering of switching genes. And apart from the individual genes, we can order the pathways along the trajectory as well, and also compare the regulatory events between two trajectories, such as the bifurcations. Then back to our trajectory of naive to dysfunction state. We applied gene switches, and here is the results. We observed that the cells are losing naive markers, such as TCF7 at the early time point, followed by acquiring dysfunctional markers, such as LAG3, CTLA4. And remarkably, we observed this transcription factor, SOX4, which is overexpressed in the dysfunctional clusters compared to the rest of the cells. For this observation, we first validate using computational way, which we check in two external data sets. One data set consists of 11 HNSCC patients, and the second one consists of the squamous cell carcinoma patients. So in both data sets, we observed the overexpression of SOX4 in the dysfunctional compartment. Then for the second data set, it also provided the information of before and after anti-PD-1 treatment. So we observed the expression level of SOX4 was reduced after PD-1 blockade, where there is expected reactivation of tumor-targeting clones and reduction in the dysfunctional phenotype. Then for the experimental validation, we performed RNAi-based knockdown on activated PBMCs and the TILs derived from our primary tumor. So in the middle for the PBMC data, if we look the red dots, which is the SOX4 knockdown, it resulted in a reduction in the dysfunction and CD39 positive and PD1 positive dysfunctional populations. Similarly, in the TILs, we derived it from 4 patients, and if we focus on the putative tumor targeting CD39 positive subpopulation, SOX4 knockdown results in a reduction in the PD1 TIL subpopulation. So all these results implicate that downregulation of SOX4 reduces CD8 T cell dysfunction in our patient. So in summary, single-cell data allows us to identify the heterogeneity of cell states in T-cell dysfunctional evolution, and we developed computational tool gene switches to identify the key gene changes during cellular transitions. And importantly, we identified a regulator, SOX4, whose down-modulation could improve the immunotherapy by reducing T-cell dysfunction in head and neck cancer. Okay, with that, I would like to thank Prof. Owen Rackham and Prof. Gopal Yen for the optimal support during my PhD journey, and also the postdoc John for helping with the development of gene switches. And the postdoc Hongshen and Lista for all the experimental work. Yeah, with that, thanks for your attention, and I'm happy to take any question.
Thank you, Elaine. I see we have a question from Christine.
Lovely talk. Elaine, thank you so much.
Thank you.
My question is, you're looking at absolute on-off of these gene switches, but do you ever see isoform switching? Is that something you can measure with your method?
Sorry, the switching of?
Transcript isoforms. So the gene itself may not be changing, but the product the gene is making may be different.
Oh, that is on the translation level, right?
On the transcription level. So it would be the class of messenger RNA that's being made. I think you've answered my question in saying that it's not something you've looked at yet.
Okay.
Yeah. Thank you.
Thank you.
All right. I actually have a follow-up question. Is this on?
Yeah.
Okay. A similar opening, which is like you've kind of used your model to characterize the genes into off and on state. Do you delineate the on and off distribution based on the input data set that's given, or a background reference that the user has to input or that you built into your algorithm?
Yeah, no, it's just input from the user, the gene expression matrix.
I see.
Yeah.
So since your data might cover multiple states, right? The on and off distribution might be quite mixed for the same so-called cell type as it transitions through states. So could you talk a little bit about how you deal with that, or what's the rationale behind if you don't care?
Yeah, sure. So our focus is the on and off switch during the whole process of transition that we are interested. And we observe this binarized distribution in plenty of genes in our data sets. So we can observe like 1,000 or 2,000 genes that would distribute this binarized distribution. So this is also our driven to focus on such kind of genes with clear pattern of switch on and off. And we believe this kind of genes are good candidates that we can target or say driving the transitions.
I see, got it. And I promise this is the last one. You mentioned that also the user needs to input a trajectory. So I guess there are many different ways to calculate a pseudo-time trajectory. So does it matter which one it is if they're different, or is your tool agnostic to whichever one is given?
Gene switches can take in any kind of, so long as it's a matrix of data which representing a pseudo-time ordering. It doesn't care what kind of trajectory inference methods the users are using. Yeah, so it's quite flexible in this case.
Okay, fantastic. Thank you. Let's thank Elaine one more time.
Okay, thank you.
Oh, sorry, there was a question. I am so sorry. Let's take that last question. Yeah, go ahead.
So the difference between naive cells. Oh yeah, they are basically the same because they share the same gene marker features. Yeah. Yeah, they are expressing those naive markers in the data set.
Oh, I see.
Sorry.
Oh, okay, yeah. Anyway, time is out. Thank you very much.
Okay,
Okay, thank you.